as you are able to see, the first step is completed. This is the lysis step. The next step is absorbent step. There the machine is absorbing the RNA from the reagent. Then the machine will go for the wash 2 times. Then they put the RNA in the 6th column, that is in the elute buffer. And here is all the temperature for each and every steps. Okay. Now the machine is washing the RNA. There is the 2 washes for the RNA. Machine is washing the RNA. As you see, the 3rd step is complete, and the 4th step is again the wash. This is 60 times mixing the wash, and this is how the going to wash. As you are able to see, if I make it zoom for you, that it's washing in the 3rd column. Okay, in the 2nd column it is already washed. In the 3rd column it is washing. Just see. This is the 1st column, this is 2nd column, this is 3rd column. Now see, this is the elute step. In the last step it will discard, means all the process is going to be stopped. So in the elute states, this is what the machine is doing. The machine is mixing the RNA with the elute buffer, so that we can easily take the RNA, and it will mixing the RNA in the 6th column where we put the elution buffer. I hope you understand, right? If you find any difficulties, you can leave a comment in the comment box, or you can text me in my WhatsApp number. Okay, this is how the machine works. This is 50 seconds left for the mixation. This is the last and the final step is discard. Yes, the end of the experiment is completed. We need to click the OK. After clicking the OK, what do we need to do? We need to open the door of the MISPA magenta, pull the sliding options, check the take the deep plate file, put it here, push the slide, close the door. All is going to be set. As everything is stop. Now press the back icon, then turn on the UV light, so that if there is an infection, the UV will kill the infections. Now what we need to do, we have to open the biosafety cabinet and put the dipole plate here. This is the microcentrifuge tube stand. We need to keep it here. Microcentrifuge tube. This is the microcentrifuge tube. We have to close the cap and write the IT for the patients. I am taking 2 microcentrifuge tube for the aliquot, as because I am testing 2 sample only, so need to close the cap, write the ID. Now put it in the microcentrifuge tube stand. Now what we need to do, now we have to take the 50 microliter RNA extraction from the 6th column, because I already told that we have put the elution buffer in the 6th control, so the machine is put the RNA in the elution buffer. Okay, so calibrate the 60 microliter. Take a new tube. For patient A, it's A6. A is the row and 6 is the column. Now you already take the 50 microliter. Put it in the microcentrifuge tube. Now discard it. Close it properly. Put it in the stands. For patient B, it's B6. Put it here. Now discard it. We have to discard it in this beaker. Now close the microcentrifuge tube. Put the micropipette in the stand. Now you have to dispose this deep well plate in the dustbin. Maybe you are able to see there is some liquid portion. This is the T lysis buffer and magnetic is there, and I hope you will able to see it. This is the first column, the second column. In the second column the machine is give their first wash. In the third column you will give the second wash, so you will able to see the magnetic bits color in the third column. In the first column this is the T lysis. Okay, so already dispose it. Now this is the 2 VTM sample. You have to keep it minus. 2 VTM sample is in the minus 20 degree.